Okay, so now we're going to transition into SELEX-seq data, SELEX-seq analysis. Right? And before we do that, I actually want to give a little bit of historical perspective. I mean, Harmon has already given sort of historical perspective on the building SAMs, PWMs from the first generation software, first generation in vitro binding data, and now I want to talk about SELEX-seq and how we applied it to Hox genes. So first, we'll talk about the Hox genes. They are homeodomain containing transcription factors, so they're in the homeodomain family. And these, it's a very large class of DNA binding, binding domain proteins. And they're famous because these are developmental genes, and mutations in these Hox genes cause these famous mutations, homeotic mutations during development of the embryo. Here's two famous mutations. You have a mutation in the antennapedia gene, right, causes this fruit fly to grow legs instead of antennas out of its head. And then here, the mutation in ultravithorax causes these, this fruit fly has two sets of wings instead of one set of wings. Right? And there's, up till recently, there's been a big paradox or conundrum in the field in that it's been shown by multiple people that these Hox proteins, these transcription factors, are vitally important in the proper segmentation of the fly embryo. And that segmentation is from the anterior to posterior, basically from the head to the feet, right, axis, in the development of the fly, and it's expression of these Hox genes in the different segments of developing embryo that makes the head a head and the tail a tail, right? However, the conundrum is that when two groups, the Noyes et al. And Berger et al., in back-to-back Cell papers, 2008, when they characterized the monomeric binding specificities of these transcription factors, they saw that they were all basically the same, right? That they were binding to the same sequences. So the conundrum is, well, how are these Hox proteins differentially regulating different downstream target genes if their binding specificities are the same? You have a question?
Do you happen to know how many Hox clusters you are doing as of now?
Okay, there are only 8 Hox genes in the fly, right? Now, but they're different isoforms. So if you look at isoforms, especially ultravithorax, you have the number gets big. That's right. So you have but there's only 8 genes. Now, we have a lot more. Okay, we have a lot more Hox genes, and they're vitally important also for us and our proper anterior to posterior segmentation during our development. But so the fly we're just looking at, here they are, right? And and of course there are different isoforms of ultravithorax. We'll talk about a little bit later. So the open question is, so how are these Hox genes differentially regulating different downstream targets, right? If they're binding to the same sequences. Now, an answer to this conundrum, okay, we believe a partial answer is cofactors. We've talked about earlier, previous question, right? So that the answer may be cofactors. And one common cofactor across all the Hox genes is extradenticle. And PBX is the ortholog in vertebrates. Right? And here's a crystal structure that was solved in 2007, showing extradenticle binding with sex chromosomes reduced, which is one of the Hox genes. It is heterodimeric complex binding to, this is a protein DNA complex, and extradenticle definitely highly facilitates binding to DNA. And so you have cooperative binding, and here is the homeodomain for the Hox protein, Here's the homodomain for extradenticle protein. And you can see here the alpha 3 helix, right? Binding in the major groove of the DNA. So its alpha 3 helix is interfacing with the major groove DNA on this side, and the extradenticle, the alpha 3 helix of the homodomain, is interfacing with the major groove on this side. And in addition, we can see in this crystal structure that there's also minor groove readout here. The YPWM arm, the N-terminal arm of the Hox protein, sequence-to-affinity, is actually lying across the minor groove and then also interfacing with extradenticle. Okay? And so this is what this extra interaction here, you can get extra affinity and specificity that you only see when Hox is binding in complex with extradenticle. You don't have that when it's binding alone as a monomer. Okay? And here is, this was the known sort of binding site motif for this extradenticle, for these extradenticle Hox complexes, where you sort of had this core, again it's a hexamer sequence, and the core consisted of this sort of AT, NN, AT, and then you had these flanking sequences on the two sides. So this is sort of what was known at the time we started this project. Okay? And so what we were interested in, we were saying, well, maybe, right? Maybe if we look at the binding specificities across the Hox genes, in which they're always bound and complex with extradenticle, maybe in this setting, right, in this context, maybe the binding specificities are different between different Hox EXD complexes. And maybe that's explaining, right, that's the mechanism with which, you know, these Hox family proteins are able to differentially regulate different downstream targets. So in order to answer that question, we developed SELEX-seq. Okay? And so SELEX-seq basically marries a classical technique called SELEX, the systematic evolution of ligands by exponential enrichment. We marry that with EMSA gel shift and Illumina sequencing. Okay? So let me go over this really quickly. So in our experiment, we started off with, you have a random DNA pool, very large random DNA pool, where you have, for each DNA oligo, you have a fixed left and right adapter sequences, and in the center, we have this 16 base pair variable region, right? And we just throw in our proteins, extradenticle and the Hox protein, and then we let that reach equilibrium, and then we do the EMSA gel shift, and we elute out the complex that corresponds to the heterodimeric complex. So here is, you know, here, this band here corresponds to heterodimeric complex. I think this one here is the monomer band, and then this is unbound DNA. Right? And so as we care about this, we elute this out, PCR amplify, and now this becomes now the library after one round. And then we use this library after one round to then repeat the procedure again. And then becomes round 2 and round 3, and so on. Now, classically, the SELEX method was used to basically just find the highest affinity sequence for any protein. You would do many, many rounds of SELEX. And what happens is you do enough rounds, and all that happens is the highest affinity sequence takes over the entire pool, right? And then it's easy to characterize. If all DNA oligos are just one sequence, it's easy to characterize what the highest affinity sequence is. However, we were interested in characterizing the binding specificities for our Exd-Hox complexes across a wide range of affinities, from high affinity to medium affinity to low affinity binding sites. So because of that, we only performed at most 5 rounds of SELEX, right? And after each round, we would perform deep sequencing. So we have deep sequencing results after each round of SELEX, and we also perform deep sequencing of the initial pool. I'm going to talk more about later. The reason why is because the initial pool, although ideally we want it to be random, is actually far from random. Okay? There are PCR amplification biases and DNA synthesis biases, double stranding biases that accumulate in the pool, and we have to normalize that out, right, in order to to properly get relative affinity estimates based on relative enrichment. It's sort of analogous with someone that talked about earlier this morning when you look at ChIP-seq data, right? It's sort of analogous to the fact that genomic sequence is very far from random, right? Different species, different genomes have different GC biases overall, but also on particular chromosomes. There's these GC isocores and stuff, and so you have to take that all into consideration. Same thing applies with this in vitro data. It ends up that our initial pool is far from random. Okay, and what we're going to do is we're going to use a Markov model to normalize out these biases. And in the R lab we're going to do later, you're going to look at these, see the results from these Markov models, and we're going to apply them to normalize out the biases. Okay? Where was I? Yeah, and then this is deep sequencing data that we're going to analyze in order to build our affinity models for the Exd-Hox complexes. Any questions? Yeah.
Sorry, so you only do one deep sequencing at the end? You do the cycles without deep sequencing, or is the deep sequencing the input for the cycle? So
It's just the out. So it's the output. So basically, after we sequence only a sample, a sample of the library, and then after one round of SELEX, right, then we take part of this library and we sequence that. So we just take basically a part of the library, sequence it to find out what is the composition of the DNA oligos, right, in the variable region that the protein is binding to. So let's really quickly make sure everyone's on the same page. Let me give an example of what happens, between two different oligos, and show you how this exponential enrichment occurs, right? So let's say you have two DNA oligos, A and B, okay? And in reality, we don't know this, but this is what we want to discover. In reality, oligo A has 10 times the affinity of oligo B, okay? Oligo A is 10-fold affinity of oligo B. Now, if in the ideal setting, our initial pool is completely uniform, okay? It's completely uniform. So in the library, the number of oligo A's is the same as the number of oligo B's. After one round of SELEX, right? What should the proportion of, in the reads, what should be the proportion of we see the oligo A over oligo B? 10 to 1, right? 10 to 1. Start off uniform. Oligo A is 10 times the affinity of oligo B, and we did the binding reaction correctly. After one round of SELEX, we perform the sequencing. We should see in the reads 10 times as many reads of oligo A than oligo B, right? Now, we take that library, and now use that library as input for the next round. So after the second round of SELEX, what will we see as the proportion, right, of reads oligo A over oligo B? 100 to 1, right? Everyone see that? It's 100 to 1. And then use that as input for round 3. After round 3, what will be the proportion? Everybody? 1,000 to 1, right? So you can see, that's the exponential enrichment. After every round, you see this exponential enrichment for the highest affinity sequences. See that? Okay, so now we're ready to look at how we analyze this data. So here's sort of the big method overview for SELEX-seq. This first top box, this is basically a cartoon characterization of the previous slide of how the experiments are done, right? And now we're going to look at the data. Again, we do high throughput Illumina sequencing of the initial round, initial pool, as well as the output from each one of the rounds, right? So we get our FASTQ read files, and then here is where we start doing oligonucleotide tabulation. We basically started count k-mers. We count k-mers at different lengths, okay? And from the round 0 reads, we build a Markov model of the round 0 reads to model any nucleotide biases, sequence biases, initial pool. And now, using the Markov model, we can now calculate what's the fold enrichment, right? R1 over R0, R2 over R0, and R3 over R0. Right? So this is the relative enrichment, and after each round. Okay? Now, also what we do is we take the nth root of these ratios. Right? Because we know, remember, after 2 rounds, oligo A was represented 100-fold more than oligo B in round 2, even though the affinity difference was only 10-fold. So that's why we would actually take the square root of the R2 counts over R0 to get to then calculate relative affinities from the relative enrichments from a sequence round. And then for this ratio here, we take the third root, right? That's how you go from 1,000 to 10. Take the third root. And then we have this magic black box here, where we do this lowest base regression to integrate these different relative enrichment estimates to come to a final sequence of affinity model that we want to maximize precision and accuracy. And I'm going to talk about that in the next slide so you understand what I'm saying. Okay? So the end result of this is to come up with an accurate and precise sequence to affinity model that I showed before, where we trained on PBM data, but here instead we're training on SELEX-seq data. So let me show you an example of what's going on here in this lowest space integration method. Okay, so what we are really are trying to achieve here is we want to calculate relative affinities via this normalized relative enrichment of k-mers across the different rounds of SELEX. Okay, and it's pretty simple. See, in 2008, Levine and Hamilton-Nielsen showed in their paper was mathematical analysis of SELEX. They showed that calculating relative affinities. So this is a relative affinity where you have this is the association constant for any particular sequence, right? Say 10 base, or actually these are 12 mers. So any 12 base pair sequence, its association constant over the highest affinity 12-mer sequence. So that's the relative affinity. So it's this ratio of the association constant over the optimal, the highest possible association constant. So now your relative affinities basically go from 0 to 1, right? It's just looking at the rth root of the ratios of ratios, where this is the ratio of the frequency of that oligo, right, over the frequency of the optimal, the highest affinity oligo. What is that ratio of frequencies after R rounds, right, over in the initial pool? What is the ratio of the frequency of some sequence over the optimal highest affinity sequence? Okay? And now, we don't actually know exactly what these frequencies are, but what we do is that we hope, right, that our sampling of this big pool, when we get the sequences, we hope that our sampling is big enough so that the relative counts, right, the relative counts of these different k-mers is proportional to the relative frequencies. This is where it's important that you do enough deep sequencing so that you have good confidence in that your counts, your frequency of counts is a good approximation to your ratio of frequencies. Your ratio of counts is a good approximation to the ratio of frequencies in the overall pool. And so now let me show you what happens when we take, say we do this ratios calculation using R1 over R0 data, right? And we compare that with using R2 over R0. Okay? So what's happening is we actually have two relative affinity estimates. One is where we're using So much for that pointer. One is where we're using the R1 over R0, right, ratios of the counts. And then here on the x-axis, this is where we're using looking at the square root of the R2 over R0 counts. And in the ideal world, all these green points will be on this black line, right, the unity line. We see there's a curvature. And the curvature is because there is saturation. Okay, even though we try to make sure that there's the protein concentration was far, far less than the DNA concentration in the SELEC experiment, there's still some curvature in here. So there's saturation of the highest affinity binding sites. And also, if I were to replace the x-axis with R3 over R0 affinity estimates, this curvature actually would get even steeper. So what happens is actually this saturation effect, it builds up in the library. As you look in later rounds of the SELEX-seq data, this curvature is more and more. Okay? So we want to normalize that out, basically. And we do that by doing this lowest regression. And what we want to do what we want to achieve is that the fact that we know that the R1 counts, the R1 data, right, is the most accurate because it has the least amount of the saturation buildup, saturation bias. However, the R1 counts are the lowest. You have very low counts, right? And we know, because Harmer talked about before, that the standard error, right, of these counts scales as 1 over square root of n, where n is the number of counts for any one of the DNA oligos. In the later round, we have less accuracy because we have this buildup of the saturation effect, but we have greater precision because we have higher counts. So it's by doing the lowest regression, we're trying to integrate the sort of marry the higher accuracy of round 1 with, in this example here, the higher precision of round 2. Okay? And actually, you can see here, I'm plotting here the standard error for of course the vertical error bars. That's the standard error using the affinities using R1 over R0, and the horizontal error bars, that corresponds to the standard error for using the R2 over R0, right, affinity estimates. And you can see here that the standard error bars are pretty large, right, for the R1 over R0 estimates, but you actually can't even see the error bars for the R2 over R0 affinity estimates. You can't even see the horizontal error bars. So that's telling you that that it's the R2 over R0 affinity estimates that have greater precision. Yeah.
There's saturation bias from the protein and also PCR bias from the amplification.
That's right.
What would happen if you did randomly sample, like without selection, your R0 pool and did PCR each round?
No, that's a good question. We did that actually. So we did that experiment where we label out the amplification only, right? We tried to then, try and model this increase in the pool based on PCR amplification biases. We weren't very successful. The problem is that notice that if when I looked at multiple rounds, there wasn't a consistent increase in the bias. Sometimes basically you would see going from say round 0 to round 1, there'd be some application bias, and then going round 1 to round 2, there'd be a lot less, and then going from round 2 to round 3, there'd be even more. So the problem is there wasn't a consistency in introduction of those biases, right?
By doing this, you just lump them together.
That's right. That's right. Yeah. Oh, do you guys know what lowest regression is? Anyone here not know what lowest regression is? So it's so lowest regression is it's basically local. It's a local think of it as piecewise local regression. Okay, it's piecewise local regression, and you use typically either linear or first or second order polynomial for each local regression. So, for example, what you would say is, for this window, so for this point here, you would look at this window, right? And you would find out, okay, for this window, what fit this parameter, right, to this one, okay? And what it does is that just will move all these points up to the black line. So it's piecewise local regression. In this example, we're using a second order polynomial that where our dependent variable is the round 1 over round 0 affinity estimate, and our independent variables are R2 over R0 estimates. So what we want to do is we want to maintain within a local window, we want to maintain the precision of these affinity estimates, right? But we want to normalize out the curve. So it says, let's look at this cluster right here, right? Now in this cluster right here, you can see that the error bars, right, the y-axis error bars are much bigger, right, than the range of where these points are. So if this cluster here, right, if you were to use just the R1 over R0 affinity estimates, the ordering of the relative affinities for these points, you have 0 confidence in, right? You have 0 confidence because all the green points are within the error bars of each other. Okay? So you don't trust the y ordering, the y-axis ordering of these green points. However, you can't even see the error bars on the x-axis. So you trust the ordering, the x-axis ordering of those green points. You see that? So the goal is you want to conserve the x-axis ordering, okay, of these green points, the x-axis relative affinity estimates for those green points. We just want to move them all together up to the black line. And that's what the piecewise local regression is achieving for you. Okay? All right. So here are the results. Okay? And this basically, this figure here kind of summarizes our entire paper right here. By using this method, and we did SELEX-seq both on the monomeric data and the dimeric data, heterodimeric data, we saw that just like the previous groups had published in 2008, the monomeric binding specificities were very similar, right? However, if we look over here on the right, the heterodimeric specificities were now, sex chromosomes reduced and altered by thorax, are now bound with the same cofactor extradenticle. Now we see that there are major differences in specificity, right? And they actually fall into different clusters. Here, this magenta line, and all of these are, by the way, these are nimers. So every point here corresponds to a monomeric nimer binding site. And then over here, the heterodimeric binding site is a 12-mer. So each one of these points represents a 12-mer in affinity space. For this magenta line here, all these 12-mers had the same ATTTAC core sequence. And that's the color magenta. And then where you are on this line is totally dependent on your flanking sequences. So your flanking sequences highly modulate, where you are on the line and your relative affinities, but they don't determine what line you're on. Okay? And then for the blue and the green motifs, right, they had different cores, ATTAAT and ATTGAT. So you can see for these two classes of 12-mer binding sites, sex-chromosomes-reduce greatly prefers these binding sites over UBX, right? This is the relative affinity for UBX, and this is the relative affinity for sex chromosomes reduced in complex with extradenticle. And then this red class here, which had the core sequence AT, both extradenticle and sex chromosomes reduced basically bound to these sequences almost equally, with equal affinity. You see that? And this is really the take-home message of the whole paper. And that message is that context matters, right? And cofactors matter. And when you're not binding with a cofactor, there's very small differences in specificity. But when binding with the same cofactor, you see major differences in specificity. And theoretically, we believe it's because there's different interactions with the minor groove, with the previous crystal structure, right, that I showed you. It showed the crystal structure of sex chromosomes reduced and extradenticle, that complex bound to DNA. Whereas when you replace sex chromosomes reduced with other Hox factors, right, that's going to change the minor groove interactions, which is going to change the specificities of the nucleotides that are in that correspond to that minor groove in the center of the binding site. Any questions? No? It's crystal clear? Okay. So now we perform that analysis, right, for all the other Hox proteins. And then we can summarize them with these plots here we call our fingerprint plots, or specificity fingerprint plots. So we did the exact same analysis, but now we looked at all of them. So we have labial, which is expressed in the head, all the way down to abdominal B, which is expressed in the abdomen. And we can see here that there are these major classes that we've color-coded. So red, blue, green. And then it's very interesting because we can see, say, for the red motif, so the red motif has got this TT in the center, right? You can see here that as you go from head to tail, we see this increase in affinity across the Hox family. But then if you look at the blue motif, which has got this TA in the center of the binding site, it's like it's low and then it gets high. For it to form, the sex chromosomes reduce, and then it gets low again, and so on. And then here, this is a summary heat map of this data here. And the summary heat map, what it's showing is, I realize I need to, the summary heat map is showing, here I've clustered the Exd-Hox proteins based on their affinities for these different consensus sequences corresponding to each affinity class. And we see three main affinity classes. Class 1, which consists of labial and proboscipedia. They're expressed in the head. Class 2, which consumes of deformed and sex chromosomes reduced, right? And then Class 3, which includes antennapedia, ultrabithorax, isoform 1A, and isoform 4A, abdominal A and abdominal B. Okay? And the interesting thing is that this clustering of the specificity of these Hox proteins is collinear with their expression along the anterior to posterior axis of the developing fly embryo. So that was an interesting finding. Which also, which has been known before, is it's also collinear to actually their placement on the same chromosome. So basically the clustering of their binding specificities is collinear with their expression patterns in the fly embryo, which is also collinear with their ordering on the same chromosome. Okay? So, class 1, class 2, class 3. So here's the summary. Yes?
You may have already spoken about this and I missed it, but so each of these proteins individually will have comparable binding specificities, but then when they're in these complexes with the same, complex protein, that's when they shift. What is known, and perhaps I missed it, about how those two proteins are interacting? I mean, one, there's an interaction with the DNA, but then the interaction between the two proteins, and I guess I'm maybe missing that. Like, like how, I mean, based on what we know about those, is there, like, could you have predicted some shift, I suppose, prior to looking at their...
Yes. No, that's a good question. It's basically based... You're absolutely right. The guesses, right, and what led us down this path to look at, right, this heterodimeric complex, which is extradenticle, is all from this complex, this crystal structural complex, and another crystal structure complex. There's this one, which becomes reduced, and here there's a particular binding site. Okay, this binding site actually had the TA sequence in the center. Okay, and we saw that there was this minor groove interaction that I spoke of before. There's another complex, exact same two proteins. The only thing different was the DNA sequence. They mutated DNA sequence so that there was no longer TA core. And what they saw was that there was no longer this interaction in the minor groove, right? That this was basically unstructured. And so that was sort of the hint, right? That, hey, when they bind in complex, there's cooperative binding. Part of that cooperative binding is the fact when they bind in complex, right, you have this extra recognition of the minor groove. Notice that if these two were to bind as monomers, right, you just separate them by, move this up here and move extradenticle down here. If they bind as monomers, the only interaction you have is just through the alpha-3 helix, interacting through the major groove. But here, when they're brought together, right, you still, of course, have the major groove interactions between the two alpha-3 helices. But then you also have this additional minor groove interaction.
So it's not either of these proteins or together, or either of them individually with the DNA. It's both of them together and the DNA.
That's right. So here's our summary figure for our Exd-Hox analysis. It's basically showing that the extradenticle unlocks our latent DNA binding specificities. So we came with the latent specificities. Because when they bind as monomers, right, there's no difference in the Hox specificity, but then when we throw in dimerization with extradenticle, we get this, latent specificity, and now we see large specificity differences between the different complexes, in which every Hox protein is in complex with the same extradenticle cofactor. Okay. All right. So now that's the biological story. Now we're going to talk about a little bit how we build affinity models using this new SELEX-seq data. Okay. And there's two methods. There's the method that I just described to you, which is what we call the Kamer method, right? And the Kamer method is what we published, and that's where you calculate, all 4 to the K Kamer relative affinities based on this, right, Kamer enrichment to multiple rounds. We do this ratio of ratios, right? And then we do this lowest base integration, different ratios calculations. So that's what I talked about before. The new method that we're just finished is using FeatureREDUCE. Here is where we're using the Poisson regression method. Okay? And there's major advantages of this. One is that the Poisson, the FeatureREDUCE method, what I showed before, is, we're trying to parameterize these affinities based on, first you have a positional independence model, right, SAM, plus possible dinucleotide correction terms, right? That's a major reduction in the number of parameters, a lot less than 4 to the K, right? Up here, you have you have a parameter for every possible Kamer. Whereas here, we're trying to actually, instead of using 4 to the K parameters, it's really only 3n plus 1 parameters, where n is the length of the binding site. That's how many parameters you have in your SAM matrix. Plus additional parameters for your dinucleotide dependencies if you have any. And so that's a sort of technical thing. But what's really appealing for the biologists is that by using the Poisson regression method, you only have to do one round of SELEC-seq. So you save time and money, right? Because sequencing is still pretty expensive. So we've implemented this, and now the question you have to ask, of course, is, okay, can this method, right, which uses only one round of SELEC-seq, remember you get low counts, right? You get low counts from only round one, which we know from the prior slides that the round one data, right, is less precise because it had a lot more standard error. Standard error scale is more of a square root of n, and n is your number of counts, and we have a lot fewer counts in round one than we get in later rounds. The question is, okay, using this method, how well do these relative affinity estimates match the Keimer method? That's the next slide. So, here's what I'm showing. First, let's look at the left, on the left here. On the x-axis, these are our 12 more relative affinity estimates using the Keimer method that we published in 2011. Okay? On the y-axis, these are relative affinity estimates using the new feature-reduced Poisson model. So we're using, actually the y-axis estimates are twofold handicapped. One, we're only using round one data here, and not rounds 1, 2, and 3. Okay? And there's a huge reduction in the number of parameters that we're training for. And we want to see, okay, given those, even though the y-axis, right, feature-reduced Poisson model affinity estimates are twice handicapped, how well do they match the Keimer method, Keimer model? And we can see for the positional independence model, we actually get a pretty good match with an R squared of 0.83. And each one of these points, just like the previous place, one of these points corresponds to a 12-mer, right? However, if we throw in the adjacent dinucleotide dependencies into the model, we see much tighter fit. Now the R squared is 0.92. So this is really great, because this is really telling us two things. The first thing is telling us is that there are significant dinucleotide dependencies, adjacent dinucleotide dependencies. Okay? And the second thing is that feature reduce, using just one round of SELEC-seq data and the Poisson model, is able to effectively capture a lot of these dinucleotide dependencies. So any questions? Okay. In the interest of time, I'm going to skip this. This is just where a major research area now in the field is having to do with binding modes. We talk about proteins that bind to DNA in different modes. The BZIP proteins are an example of proteins that bind different modes. And the two dominant modes are what we call the overlapping half-site binding mode and the adjacent half-site binding mode. And you see what's happening is basically columns 0, -1, and +1, right, are merging to column 0 here. And I should say that the BZIP proteins, they bind as homodimers and heterodimers, but here we're just looking at homodimer data. Okay? And a major challenge is that when you look at PBM data or SELEC-seq data, of course in SELEC-seq data, right, you can elute out your complex, so we would elute out the homodimeric complex, right? But even in that band, you would still have sequences, right, that correspond when you have the overlapping half-sites binding and when you have the adjacent half-sites binding. And they would both be the data would be confounded with both binding modes. Now, I've only shown you two binding modes here, but a lot of proteins can actually have a variable length spacer, right? So instead of just having either a spacer of 0 or 1, you could have lots of spacers from 0 to 15, okay? But the data is confounded. So we put together this convergence procedure that actually can help unblend the motifs. So you actually get truly unblended, independent affinity models for every possible binding mode. And on top of that, we also can then calculate what is the relative affinities between the different binding modes. So let me show you the example. Oh, this is just showing that this model here has a lot more specificity than this model here. This is the beginning model. If you just take the seed sequence, for the half-site, overlapping half-site model, and you just generate a PSAM, you get this. And if you take the seed here for the adjacent half-site model and build up a SAM, you get this affinity matrix. And you can see that this model has a lot less specificity than this model. That's because these two models actually are blended together because the data is confounded. So let me show you the end results of this procedure. So YAP1 is a BZIP protein, and we can see that it actually prefers the adjacent half-site binding mode over the overlapping half-site binding mode. And the binding motifs are symmetric, reverse complement symmetric, because its binding is a homodimer. So you see basically a 50% reduction in affinity for the overlapping half-site mode. However, if we look at another BZIP protein, GCN4, we see actually it prefers the overlapping half-sites over the adjacent half-sites. And there's about a threefold difference in affinity between those two different binding modes. Any questions? Okay. So here's just a summary. FeatureREDUCE. We have a biophysical framework that infers, right, affinity models from data. We want to discover the minimal set of sequence features that best captures the sequence specificity. We use robust regression, which we talked about a lot already in the past week and a half. And that's a great technique to avoid overfitting to noise. Accounts for platform-specific biases. Here, specifically for PPM, we had this artifact, right, where the protein has difficulty getting into the closely compacted double-stranded oligos for a spot. Accounts for nonspecific binding. We capture energetic dependencies because of nucleotide positions. And I didn't show you, but we also can use the same framework to then also capture energetic costs for any nucleotide insertions or deletions in the binding site. Multiple binding modes, I just showed you. And then we also have the relative affinity can be estimated between different binding modes. And then finally, the two different types of data. We have the real valued, roughly normally distributed fluorescence intensities from PBM and HITS flip, Monomer. I didn't have time to talk about HITS flip and Monomer. We're going to talk about it later. And the integer value of responder sensitivity counts from high-throughput sequencing and vitro binding assays. And to thank, of course, Harmon and everyone who was on the lab and collaborators, Richard Mann and Matt Slattery. Matt Slattery did all the experiments for the SELEX-seq Hox data, and Matt and Tim Hughes. And thank you for your time. Yeah.
So did you do the one-round SELEC-seq with the Extradenticle data? Was it able to pull the heterodimer data? Was it able to pull out the differences between one binding mode and the other binding mode?
Yeah. So this these binding modes for GCN4 and YAP1, these are actually built using PBM data. So these are using PBM. Now, this analysis, this is using SELEX-seq data.
Yeah. So did you do this method with comparing the was it SCR by itself and SCR with Extradenticle?
Yeah.
And was it able to pull out the difference between those two modes?
Yeah. Because the R squared is so close, you know, if you repeat this analysis using the new SON, right? If we repeat all the analysis in the cell paper using our new SON model, you get the same answer because you said such a great fit.